Assalamu alaikum and good morning, and welcome to this morning's session of Breakfast at UMHealth. My name is Azlina, and I will be moderating the first session. Without further delay, let me introduce the first speaker, Associate Professor Dr. Mohamad Fazril, Mohamad Razif. Dr. Fazril is a member of the Department of Molecular Medicine, which he joined in 2014. He completed his undergraduate education at Murdoch University in Perth with a double degree in biomedical science, molecular biology, as well as in forensic biology and toxicology. In 2013, Dr. Fazril received his PhD from the University of Western Australia in the field of synthetic biology. In 2017, he joined the Medicinal Mushroom Research Group and has great interest in studying the genomics, transcriptomics, and proteomics of medicinal mushrooms. His current research focuses on the expression of novel proteins found in medicinal mushrooms and the identification of their bioactivities in vitro and in vivo. The identification and validation of bioactive molecules from nature and their activities paired with clinical studies will enable for the future development of natural compounds for pharmaceutical prevention of diseases and health maintenance. Today, Dr. Fazril will be sharing with us some of his research work with the title of Development of Clean Cosmeceuticals Using Medicinal Mushrooms. Can I invite Dr. Fazril?
Hello, good morning. All right. So today is going to be a sort of like a passion project, fun project that we at the Medicinal Mushroom Research Group have sort of like thought about over, I think it started around when we were in lockdown. And that's when I think me and Prof, who is the founder of the Medicinal Mushroom Research Group, sort of had this discussion. We both were passionate about skincare. And based on the research that we've done in the lab, we thought, hey, we can do something with this. And this brings us to the talk today, which is the development of clean cosmeceuticals using medicinal mushrooms. There are two things that I guess we sort of like have been terms that we've been hearing of late, and it is nutraceuticals and cosmeceuticals. So nutraceuticals are any substance that is a food or part of food that provides a medical benefit or health benefit that can be consumed physically, so we take it through orally for the prevention and treatment of disease. While on the other hand, we have cosmeceuticals, which are generally cosmetic products that contain bioactive ingredients that have some form of pharmaceutical action on the skin. And this term was actually derived back in 1964. So both these products, they contain basically bioactive ingredients that can be anti-aging, anti-inflammatory, to treat pigmentation and also to improve the complexion of skin. And I think over time, coming out of the lockdown, this was a very popular, I think people just had time to sit at home and really think about what they're putting on their skin, what they're actually eating, and being a bit more conscious about what they put in. So there's been a lot of products that are like functional foods, dietary supplements, which we have been like supplements we've been taking for like a really long time as a society, and now looking more into what we can do for our skin. So just touching a little bit on nutraceuticals. So these can be, we've heard of probiotics, prebiotics. We take our vitamin pills. And basically, all these supplements have health beneficial. They are health beneficial products, generally from food itself, that may have been isolated or even enriched for our own consumption. And then what I want to focus on today is cosmeceuticals. So cosmeceuticals, again, as I mentioned, is something that we apply topically on our skin. Generally, they can consist of protein or peptides. We have our hydroxy acids for hydration, also retinoid, vitamin A, all encompassing to make us look very beautiful, very radiant and useful, but also can be used to sort of treat certain skin afflictions, and we'll get into that a little bit more later. So cosmeceuticals are applied topically, and given that the epidermal layers of the skin, like blood vessels, there's still a lot of research that needs to be done because not all compounds can penetrate through the different layers of our skin. And now you see products that are being developed, for example, like nanoparticles, encapsulation. You're looking at a simple compound now is, for example, hyaluronic acid. And they're having multi-molecular size hyaluronic acid to have different levels of penetrative action. Of course, hyaluronic acid we know is good for hydration, but we also know that there are some people or individuals that may be sensitive to certain molecular weight hyaluronic acid because it penetrates too deep and they actually have reactions. So an issue now surrounding cosmeceuticals is that, I mean, for big companies, we don't really see the research that goes behind their product development. And here at the Medicinal Mushroom Research Group, we really do want to eventually have a product that, that we've done all the in vitro and in vivo trials, that we also have had our clinical trials. So we actually have a product that we have really strong scientific evidence for it. So I'm sure many of us here have heard of this like active compounds. You hear about ceramide, which is good for skin barrier. Also niacinamide, which is a precursor to NAD, which is our energy molecule. And what we want to do is basically you need all these active compounds to sort of support skin health, be it vitamin C and E, peptides, ceramide, all depending on what we want to sort of like treat, an issue that we want to treat. But research has shown, if you go through literature and you see research, although these compounds, they are synthetically derived and also naturally derived, how they also perform as a compound itself actually does vary. It really depends on what the source of the compound is and also how this compound was extracted. You would think, like, if you take, for example, like ceramide, I mean, if you say via water extraction or something that is more lipid soluble, you would think that, oh, it's the same compound, it should perform the same. But research has actually shown that depending on how you extract this compound, it does affect the action, and it may be coming down to more of the slight small changes in the molecular structure. And there is just not enough research out there to actually, no one is actually looking at these structures, these chemical structures, like, closely. So, I mean, talking about cosmeceuticals, we were all once young, and now we are constantly being exposed to a lot of environmental factors, UV radiation, our air is not as clean as it is, so there's a lot of contaminants out there, and this all is going to add up to our skin aging much more rapidly over time. So when you're talking about young skin, what's going to convert young skin to aged skin is more extrinsic and intrinsic factors. So intrinsic is more like just biological aging as a process in itself. Also, what we as a organism has sort of gone through life, like a stressful life versus someone that's more relaxed, it does really play a big role in how rapidly our skin ages. And it all comes down to the increased production of like reactive oxygen species. And we also have a decline in some enzymes like hyaluronic acid synthase, also our extracellular matrix proteins that's it declines over time. So we're just trying to reduce the rate of decline. We cannot really avoid it, I think, but we want to reduce that rate of decline. So when there's increased reactive oxygen species in our skin, we generally do see a decreased thickness and in our skin. The quality is not the same. We can get a little bit more dry and get more dull. And yeah, we lose collagen, we lose elastin. So all of this adds to the process of aging as we go on through our lives. So in terms of mushrooms, so in the medicinal mushroom research group, we do work on a handful of mushrooms. I think at this point we have about 7 or 8 mushrooms that we're working on. But on this earth, so far there's about 14,000 known species of mushrooms, and it has been estimated that there may be about 140,000 species. So there's many, many more that is yet to be discovered. And if you were to go on NCBI and look up research on mushrooms, there are generally a select few that we always see popping up, like Ganoderma lucidum or Linksy. We have Cordyceps sinensis, which is what we work with as well in our labs. So mushrooms, they are the superfood. There are a lot of benefits when it comes to mushrooms. Not only is it low in calories, it has really good content, nutritional content, okay? It's low in calories. They can be high in selenium, potassium, and vitamin D. And they actually contain a lot of glycoproteins and potent antioxidant enzymes. They also have certain chemicals, or known as microchemicals, that are quite unique to mushrooms. This can come in the form of alkaloids, or lectins, or even like saponin. Then, based on research, they do have certain bioactivities. Many of them do have antioxidant activities. Here in the lab, we have shown that certain species of mushrooms do have anti-cancer properties and also anti-diabetic properties. For example, like saponin and tryptophan, research has shown that these compounds from mushrooms have been known to lower cholesterol, for example. So now it's just trying to harness and do more research about it and see how we can apply it in a medical setting. And for the purpose of this talk, what can we do? What can we use these mushrooms for in terms of our skin and skincare? Using its anti-inflammatory properties, anti-collagenase properties to help make us more radiant, make us look youthful for longer, but also to treat some skin afflictions in a more natural way. So this is typically a very common, typical structure of a mushroom. It has a cap at the top, it has a stem, and also at the bottom part. Sorry, can you see my cursor? I hope you can. At the bottom, we have the mycelium as well. And we have mycelium and the sclerotium at the bottom. So depending on the mushroom, the bioactive compounds can actually be found in different parts of these mushrooms. What you need to do is you need to study, not only look at its physical parts, but also the growth period, because for certain mushrooms, the bioactivity is high, or the content of this bioactive compound is high, be it just before it sprouts, or sometimes even after. And there has also, like, when you culture it in the lab, or when you grow it, sometimes there are the culture broth, and people actually have been using this culture broth and studying this culture broth because they found that there's also a lot of bioactive compounds that are being released into this media that can be used for medical purposes. So I'm just going to walk you through the workflow that we usually do in our lab, in mine and Prof's lab. So we're just going to look at some safety evaluation studies, and we look at nutritional studies, and also we touch on a little bit of like the translational studies that have been done. So before we do any work, usually we would generally go through a safety evaluation study. This includes like a subacute toxicity study that usually lasts about 28 days. Then we do a chronic toxicity study for about 180 days. We check for the in this mice. We check for whether it affects their fertility, whether any of the organs are affected by these by consuming these mushrooms. And thankfully, that's far all the mushrooms that we work on, they're all safe to be taken orally. All right, then we go on to nutritional studies. So one of the mushrooms that we work on specifically, and something that is going to be focused on in this talk, is our tiger milk mushroom. And it's actually it's called Lignosus rhinoceros, and it is found in Malaysia. And so far, there are three Lignosus species that are found in Malaysia. That is rhinoceros, cameronensis, and tigris. But a lot of work thus far has gone on with Lignosus rhinoceros. So we usually do a study, compare it to the wild type, because the one that we have now, it is actually in partnership with our industry partner, Ligno, who actually supplies us with our material for us to study, and it's usually supplied as a dried powder, in a dry powder form. So we perform nutritional studies. How does it differ with our the wild type species? We also generally do a cold water extraction. I mean, there's different types of extraction processes that you can do, but in our lab, we would only work on the cold water extract. And from that, we would fractionate it even further to high molecular weight, medium molecular weight, and low. And also, then we also look at the nutritional content within each of these fractions. Then something that is usually easy to move on into is our antioxidant studies. So we know that mushrooms, they can have, DPPH radical scavenging activity, methyl chelating activity. So we want to see, we'll use this extract and usually test them across a range of antioxidant assays to see which one it is most active in. And you will find that different fractions will actually have slightly different bioactivities. And of course, why do we need it? Is, again, we don't want, anti-glycation end products forming anything that is going to, hopefully we want to reduce our reactive oxygen species levels in our bodies. So we want to make sure that we can have high amounts of these really good compounds that we're taking. We also actually, with a faculty grant that we got, the MRG grant that we got about two to 3 years back, we were able to, in collaboration with Dr. Amira Hajira in pharmacy, we were able to actually do a project where we had a diabetic rat model, and we were able to supplement these rats with two types of mushrooms, our tiger milk mushroom as well as our Cordyceps sinensis. And we found that, these rats had, decreased plasma He concentration. They actually had improved mitochondrial function, increased ATP. So in this particular study, the Cordyceps, actually, if you look up Cordyceps, people have sort of identified the bioactive compound and named it cordycepin. But for our tiger milk mushroom, we have a very small short list, maybe like 3 compounds that we are sort of like really trying to focus on, and trying to really kind of determine and figure out its structure and its actual the range of its bioactivity. And hopefully in the near future we'll be able to, like, I guess, Prof, we can name this compound, this bioactive compound in this mushroom. Yeah, that is the goal. Keep on working. So other things that we do is we look at the immunomodulatory activity of these mushrooms across various cytokines and how they're able to affect or change these levels. And even from here, you can see, just the small table showing IL-5, IL-6, MIP2, and CIMP1. and how these different cytokines actually aid in different parts of our immune system. For example, TIMP1 is involved in tissue remodeling, wound healing, and also to promote growth. So obviously, to have these, depending on these cold water extract, high molecular weight, medium, and low. And as you can see clearly, that each of these fractions, or the pure extract in itself, does have different varying bioactivity. So it depends on what you specifically want to do and what are you specifically targeting, and then that's the fraction that we would want to work with. And then we have also anti-inflammatory activity that we'll look at. So we would generally just test cold water extract, high, medium, and low, see how these fractions work. Do they have a strong standalone action, or do they have a synergistic effect between the fractions as well? And then, so that is more on the bioactivity part. We also do a lot of really groundwork on trying to understand the species of mushrooms based on their genome, transcriptome, and proteome. So recently we just published a article here on the genome of the Lignus tigris, because it has not been done before. And then now we have our students working both on the transcriptome and the proteome, really to understand what RNAs are being expressed, and are these RNAs all fully being translated into proteins. And of course, again, as I said, this when it comes to this, depending on the age of the mushroom, the growth period of the mushroom, the media that it's in to grow these mushrooms, they all will, of course, be expressing different types of RNA and expressing different types of proteins. And just thinking about that, it's like it is truly an endless amount of research that you can just do on a single mushroom if you think about it. Again, mushrooms are one of those things. Now they're talking about fortifying mushrooms with, if you say you need a mushroom that's enriched with zinc, and all you have to do is, or enrich with iron, put the zinc in the media, and then you culture it, and the mushrooms actually will absorb it in, and you will have a iron-rich or iron-fortified mushroom. So that's why it's a superfood. A lot of research is really going into this field when it comes to our fungal friend here. And of course, talking about genome and transcriptome, we also have proteome, looking at how, within, for example, our Lignosus family, we have our tigris, cameronensis, and rhinoceros. How are they distinct from each other? Do they express the same amount of protein? And we actually have shown that they don't. They are not, although they are related, they are not similar in terms of their expression. Even their bioactivity is slightly different as well. So why we need to know all this is eventually, we can identify and basically characterize novel proteins that can be found only in this particular species of mushroom. So looking at skin and aging and diseases, one of our passion projects, and we actually been trying multiple times to get a grant to really get this off flying, is we wanted to deal with patients with eczema. Even I am someone who actually suffers from eczema, and at present I'm using like steroidal creams that are steroid based actually to get rid of it. But prevalence of eczema and skin conditions can affect about 20% of the population, and sometimes steroid creams are the only ones that are available right now to sort of alleviate the symptoms of this condition. But we know that there's long-term health effects with prolonged use of steroids. Sometimes there's also fragrances in the cream. So I think there is a gap here that needs to be filled, and that is to develop a cost-effective topical treatment with minimal side effects that can sort of alleviate the skin condition. So I've talked a little bit about aging and skin, but everything, as I said, leads back to the increasing reactive oxygen species. And again, obviously, there is a downward pathway that we all have to look at. We have to also deal with the inflammatory response that happens. When there's inflammation, you typically get collagen degradation. So I'm going to say, I already can see on my skin here where my eczema is. It's not looking too happy. And also you can see the quality where I always have these outbreaks. The quality of the skin there has actually deteriorated. So this is something that really needs to be addressed. So in terms of mushroom skincare, there are actually many, many mushroom skincare products out there already. And here are some of the examples that I've shown. Again, we usually get common mushrooms coming in like Ganoderma lucidum that we eat, but also now being incorporated into skincare. We have even cordyceps being used in a product in Japan. And it's for moisturizing and also suppressing melanin production. So it's about time that we in Malaysia get into this research right here and not miss this opportunity. It's been all over the news, especially since this coming out of the pandemic. Mushroom is the new hot product when it comes to being used in skincare formulation, but we want it to be more on the clinical use, also to make sure that we have a good, solid scientific data to support the claims that this product that we're developing will have. So generally, when it comes to the development of a product, of course, firstly, you have to identify the mushroom species that you want to use. Next, you want to also, once you have that, you want to identify what is the source of the active ingredient. Again, where is this active ingredient coming from? Is it coming from the cap? Is it coming from the sclerotium? Is it coming from the stem? Then the next thing that as researchers we have to think is, how are we going to prepare this material? Do we want it fresh, or do we want to work on a dried product? And actually, they are both absolutely okay, fresh and dry. It's just that for dried mushroom products, you may lose certain compounds along the way. Then at the extraction method, whether you want to have a solvent-based or a water-based, and as I mentioned in our lab, we always use a water-based extraction method here. And this in green, identification of active compounds. So I think for many current products out there, this is it's not a mandatory, it is an option for them to actually, if they are interested in actually trying to figure out what their active compounds in in this mushroom is, they would go through this process and put the extract through either some form of chromatographic technique to identify the compounds. Then what we do is we have our in vitro experiments, where we look at antioxidant potential, its anti-inflammatory potential, whether it can heal wound healing activity as well. And something that is missing from a lot of products that are out there right now is the clinical trial. So we have a lot of products, but we don't know, do they actually work? And this is something that I think, as at the Medicinal Mushroom Research Group, really, really hold close to our heart. We want to make sure that we can say that the product actually works, and this is the research and the science and the results that we have for this product. So what you'll be looking at is whether it is a hydrating product, whether it actually, again, it can be, skin brightness, skin elasticity, whether it can improve that. That makes it is hypoallergenic as well, and be able to even prevent things like UV damage. So I'm just watching for the time. I'm just gonna quickly go through this section of it. So certain things that you want to look at, and I'll show you some research that people have done. Obviously, you want a product that can reduce or or inhibit collagenase and elastase, something that's going to obviously reduce the elasticity in your skin. And also, how does it actually hydrate your skin? Is it more, emollients typically are more oil-based, but is it an occlusive or a humectant? If you really look at the back of your skincare product, and sometimes, you say hyaluronic acid, sometimes it tells you what extract, you can see a mushroom extract. A very simple example, a very common mushroom used as a humectant is actually snow fungus, the things that we eat in our dessert, the white snow fungus, because they hold a lot of moisture. I think like 90% of its body weight is moisture, and it's because it's rich in hyaluronic acid. So that is what people have been using to put in their cream as a humectant. If you didn't know that, yeah. Okay, so I'm just going through some published research that's been shown in the past. Mushrooms have polysaccharides, terpenoids, and these are very well-known compounds that have good anti-inflammatory properties, and they do target certain markers like the interleukins and your cyclooxygenases. So that, again, depending on the mushroom, they target different types of markers. And then looking more onto the skin side, tyrosinase, and also looking at melanin production. So Ganoderma lucidum, they have identified this compound called Ganoderma nodiol. All right, that is the active compound in Ganoderma lucidum. It is able to inhibit the activity of cellular tyrosinase, and also because it's able to do that, it actually decreases melanin production. And then other things, collagenase, again, something that you want to prevent, reduce the destruction of a collagen in our skin. We have 4 types of collagenases in us. There are 2 types of gelatinases. There's 24 genes encoding metalloproteinases, everything that is just gonna basically disrupt and reduce the elasticity and the youthfulness of our skin. Listed down here, we have a few research with various types of mushrooms and how they are able to inhibit collagen, to inhibit collagenase, inhibit elastase activity, and also reduce reactive oxygen species. And this particular page here is just looking at the hydration activities of these mushrooms. There's one, yes, as I said, snow mushrooms, about 80 to 90% of it is just water, and because it has high amounts of hyaluronic acid. We also have the wood ear mushroom, auricularia, which has been shown to be pro-collagen and also enables the expression of hyaluronic acid synthase in cells. So this is something that would be very good and we would want to incorporate into our formulation. So looking at our research background of our lab, we're coming close to the end of the talk. Prof actually initiated this very early on, back in 2009, into research with these medicinal mushrooms. And as I mentioned, I came in around like 2015 into the lab. So throughout this time, we've looked at, as a whole, as a lab, safety evaluation, antioxidative properties, anti-inflammatory properties. We've looked at bioactivities. We've done, I think, since I joined, we were able to do 2 clinical trials already, one on breast cancer and also one on asthma patients using this mushroom. There we also hold a couple of patents. 2 of them are listed here. And as I mentioned, this is mainly with Lignocystis rhinoceros, but we have a few small ongoing projects, preliminary studies on 4 other medicinal mushrooms as well. So in terms of the proof of concept of this product that we want to develop, we know that our in vitro results show that, it has anti-inflammatory properties because it inhibits TNF-alpha production. We know that the extract itself has strong antioxidant properties, and it's able to modulate our cytokine. So we hope that we are able to also collaborate with other institutions to sort of develop a mushroom-based, to create a mushroom-based product that is, good scientific evidence to validate its efficacy as well. So this is just research that has actually come out of the lab. So this was back in 2022, where Prof and our collaborator, Dr. Yenny from Massachusetts University, were able to publish this piece of work looking at the in vitro inhibition of melanin formation and enhancement of collagen production using the cold water extract from the mushroom that we work with, Lignostis rhinoceros. So the results thus far has been quite promising. They found that through gas chromatography, there are many cosmeceutical compounds that can be found, including squalene and ascorbic acid. Then they were able to show that at 10% concentration of volume per volume of our Lignostis rhinoceros cold water extract, that they were able to inhibit melanin production by up to like 50. Yes.
Can I suggest that you quickly wrap up your presentation?
Yes, it's literally like the last 3 or 4 slides. Yeah. Yeah, to inhibit melanin production. Same for it being able to downregulate the expression of melanogenic enzymes. So you want to decrease melanin production, yeah, for brightening effect. That the cold water extract was also able to promote human pro-collagen type 1, so at 12.5%. So it's something that we would want to maintain the structure of our collagen. And as I mentioned before, we are actually performing some preliminary studies on 4 other medicinal mushrooms, and we're looking to eventually formulate something, a multi-mushroom product. So over my sabbatical, I was able to, last year actually, I was able to attach myself to Tosongku, which is one of the biggest, companies that does a lot of actually skincare, and sell a lot of skincare. You could go to Watson's. Unknowingly, you actually would have seen their products. They are currently based in Uptown. I was able to also see how products are made and learning about formulation, because I'm not a chemist, I'm a biochemist, but I wanted to know more what goes into, stability testing, what goes into knowing how you want the product to feel. If you want to feel oily, powdery, silky, so that those are things that actually happen in their little product development lab. And I also was involved in a lot of marketing meetings, interestingly enough, learning how, most importantly, it doesn't matter how good your product is. If you cannot market it and you cannot make it sexy, it's not going to take off. So that was, I think, coming from a science background, that was a little bit of a shock to the system for me. So it was good to learn that. So hopefully in the next 3 years, we will be able to actually develop a skin support product using our medicinal mushrooms that we work on in the lab, and that we are able to evaluate the efficacy of this product using in vivo and also clinical trial studies, and yes, be able to make something that's more sustainable, that is safe, and that is natural. So this is my lab. Our lab. Prof is here, and the students that we have, our PhD students and master students. Not all are featured here, but that's everyone. We have Minjia, Prof, we have Boon Hong, and also who else? Oh my goodness, I'm people's name. Yeah, who's working on asthma? All right, thank you so much. I'm so sorry I went a little bit over time. But yeah, I hope you enjoyed this. Something different, I hope. And if you're interested in working together, do email me or Prof. Funshini here at the Department of Molecular Medicine. And yeah, thank you so much.
Thank you very much, Fazril, for the very interesting talk. I'm conscious of the time. So what I will do is, if any one of you has any question to ask, I invite you to type in the chat box, and Fazril will be online to answer your questions. So with that, I thank you and I pass you to the next moderator.